Mitochondria are rod-shaped organelles that are found in cytoplasm of eukaryotic cells. It consists of double membrane, which is outer membrane and inner membrane, leaving a small space between them called intermembrane space. The outer membrane has larger pores to allow passage for ions, molecules, and small proteins, and this membrane is made up of proteins and phospholipids. Inner membrane flow into many layers to create a structure called cristae. This folding increases surface area for biomolecular reactions. The inner membrane is full of proteins, which aid the movement of electrons across the respiratory electron transport chain. ATP is synthesized and produces energy. Dysfunction of mitochondria is caused by mutations, either in mitochondrial DNA or nucleus DNA. A parts of body commonly affected are brain, liver, heart, and muscles, which have high energy demand. Among 4,000 people in USA, one individual is suffering from mitochondrial genetic disorder. Every person with mitochondrial disease is affected differently. This is because of the different combination of mitochondria that are working and non-working within the cell. Mutation in some of the mitochondrial genes can cause leak syndrome. Most of the genes are involved in the process of energy production in mitochondria. For example, MT-ATP6 is the most commonly mutated mitochondrial gene in leak syndrome. It provides instructions for enzyme production in oxidative phosphorylation. The other genes provide instructions for making tRNA molecules, which are essential for protein production within mitochondria. Many of these proteins are important in oxidative phosphorylation. Therefore, mitochondrial genes mutation that cause leak syndrome will impair oxidative phosphorylation, which can lead to signs and symptoms of leak syndrome. Leak syndrome is a neurological disorder which is characterized by progressive loss of mental and movement abilities. It results in death within 2 to 3 years, usually due to respiratory failure. Leak syndrome usually appears in infancy or early childhood. The affected children may experience delayed development, problem with movement, muscle weakness, and difficulty in breathing. Leak syndrome can be inherited by the mutation of mitochondrial DNA and nuclear DNA. Those caused by mutation in nuclear DNA may follow an autosomal dominant, autosomal recessive, and X-linked inheritance. Those caused by mutation of mitochondrial DNA can appear in every generation of family and can affect both males and females. They are transmitted by the mother because only X cells contribute mitochondria to the next generation. If the mutation is from the mitochondrial DNA, this inheritance can be blocked before it's passed on. One approach to prevent mitochondrial genetic disorder like leak syndrome is called mitochondrial donation. This technique is to combine the nuclear DNA from an affected X cell with healthy mitochondria from another woman. The embryo will then have nuclear DNA from original mother and father, and mitochondrial DNA from the X cell donor. Such potential infants are called pre-parent babies. This new technique may be a controversial approach, but techniques like this could finally combat this devastating disease.